Okay, so the first chapter is chapter 17, which is special senses. We finished 1 to 16 in AP1, and now this is 17 special senses. What are the special senses you should know from 233? Do we have 5 or 6 special senses? 5. What's the 6 special sense? 6 sense? 6. There is no 6 special sense. It's only in the movies, right? It's in the movies. Predicting what's going to happen in the future. This is not physiology. We have only 5. And what are the 5 special senses? Olfaction, smelling, vision, seeing, hearing, taste, and what's number 5 that we always forget? Equilibrium. So these are the 5 special senses that we will be doing. The first one will be olfaction, and olfaction means smelling. The olfactory organs, the organs that we use to smell, are the olfactory epithelium, and underneath will be the lamina propria, which is a basic layer underneath. Does anybody know what is this? What is this? And what is this circle? What is this color? The off-white beige color? Is it cartilage? Is it bone? Is it muscle? It's bone. Always in the lab, when you see this color, it's always bone. Okay? So this is obviously the olfactory. Do you remember the cribriform plate? The perforation in the base of the skull? Yeah, this is the cribriform plate of ethmoid, and this is where the olfactory fibers, which start in the olfactory epithelium, you will see the olfactory receptor cells from which the fibers will go through the skull, the foramen, the cribriform plate of ethmoid, and they are going to go up, making the olfactory bulb, olfactory tract. What do you call olfactory bulb and tract together? Olfactory bulb plus olfactory tract. What do you call it? Olfactory nerve. Okay? Whether we did 233 or not together, I will refresh your information anyway. Okay? So as we go, we will refresh our information about certain parts from 233 if it is relevant. But this is what is meant by the olfactory nerve. It's not the same thing. Olfactory nerve is 2 parts: olfactory bulb and olfactory tract. Together, olfactory nerve, which is nerve number what? 1. Okay. Everything evaporate. 233. What do you mean by 233? It's AP1. Oh, okay. That's fine. Usually, if it is in the secondary memory, there is a primary memory. It's all gone. Secondary memory is something that needs some refreshing. So if you hear it again, you will remember it. Don't worry about it. So we have the olfactory receptors. These are the receptors that will pick up the signal for olfaction. There are also olfactory glands, and the olfactory glands will secrete olfactory secretions that will coat the surface of the olfactory organs. The olfactory organs are basically the nose, the nasal septum, and the epithelium inside. These are the olfactory organs. Receptors, the olfactory receptors are highly modified neurons. So this is the origin. These are neurons that got modified, highly modified, to do the function of picking up the olfaction signal or the smelling. How do we pick up the olfaction, or how can we smell? Everything that we can smell will send some chemicals. These chemicals, dissolved chemicals, will go in the nose and it will stimulate the olfactory receptors, creating a signal that will go through the olfactory nerves and go to the brain. This is how it goes. So these are dissolved chemicals that stimulate the olfactory receptors. And the axons of these olfactory receptors that are leaving the nose will go through what? Olfactory receptors, the receptors for olfaction. This is in the nose. It will pick up the signal, which is dissolved chemicals. And then the fibers, the axons, will travel to enter and go to the brain. Travel through what? How does it cross or go through the skull, the base of the skull? Through cribriform plate of ethmoid. Everything was a good vacation. Everything is gone. Cribriform plate of ethmoid. This is in the base of the skull. These are these foramina in the base of the skull where the fibers go through, penetrate to go to the brain. Cribriform plate of ethmoid. So the olfactory fibers will go to the olfactory pulp, from the olfactory pulp to the olfactory tract, and the olfactory tract will go to the cerebrum. Which part of the cortex is responsible for olfaction? Anybody remember? That was 233. Don't worry about it. AP1. Anybody remember? You remember the insula? Do you remember the cortex? Frontal, occipital, parietal, temporal, and what's the fifth one? The insula. So the insula is the fifth one that's responsible for olfaction and gustatory. So again, you're smelling something. There are chemicals that come from this something, whatever it is. It's food, it's flowers, it's anything. There are chemicals that will go, dissolve, interact with your nose, the epithelium, stimulating the receptors. From these receptors, the fibers or the axons will penetrate the skull, go through the skull from the cribriform plate of ethmoid. They are going to the olfactory pulp, olfactory tract, and they will go to the cerebrum, which is the olfactory cortex, and this is when you can tell what are you smelling. This is one way. The other thing is there are some emotions that can accompany the smelling. You can smell something that's bad and you react, what is this? That's bad, right? Sometimes there is something that smells good, so you react to it. There are some emotions that accompany the smelling, and these emotions are coming from the hypothalamus and portions of the limbic system. So do you remember the limbic system? Limbic with M is responsible for function of the limbic system, emotion and memory. If you remember that, emotion, M, limbic, M, emotion and memory. Limbic, emotion, memory. These are the functions of the limbic system. So this is responsible for emotions. So that's why you smell something bad and you feel bad. You smell something good and you feel bad about it. Bad and you feel bad about it, right? This is coming from limbic system. So not only the olfactory nerve, it's not only reaching the olfactory cortex, it also sends the signal to the limbic system. And that explains your emotional reaction to the stimulus that you are actually smelling. This will not go through. This will go through the hypothalamus and portions of the limbic system again. It will not bypass. It will not synapse in the thalamus. Olfactory discrimination is when you distinguish different types of chemical stimuli that you're actually smelling. There is a good or considerable amount of turnover, so we always lose and renew our olfactory receptors. We always lose it and renew it, always lose it and renew it. This is a turnover. And the number of olfactory receptors, just like everything in the body. When we get older, everything deteriorates, right? Everything, including the olfactory receptors. Everything goes down, you name it. When we have any question, when you think about what happened in elderly, just pick everything deteriorate. Olfactory receptors deteriorate, memory deteriorate, cerebrum deteriorate, everything deteriorate. So what happened to the olfactory receptors? The numbers decline, deteriorate. Now the taste. Same thing. We'll start with the receptors. What are the receptors? These are called the gustatory receptors. Gustatory or taste is the same thing. Like smelling and olfaction is the same thing, right? So these receptors are clusters in the taste buds. So what are the taste buds? The taste buds are clusters of gustatory receptors. And these taste buds will be associated with the lingual papillae. These are the different types of the taste buds. There are some of them are fungiform. Fungiform means looks like a fungus. Filiform. And the third one is circumvallate. It's not appearing here. Circumvallate, the third one. So we have circumvallate, we have fungiform, we have filiform. We have three different types of taste buds. This is all depending on the way they look like. Does it look like a fungus? Does it look circumvallate? Or it is filiform? So we have three types: filiform, fungiform, circumvallate. The filiform does not contain taste buds. So it's not doing the function. It's not helping in the gustatory or the taste. It's just provide friction, just moving around the chemical signals. The fungiform will contain 5, and the circumvallate will contain about 100. So obviously, the most important one of the taste buds are circumvallate. So the circumvallate contain hundreds. Fungiform, a little bit. Filiform, nothing. Does not contain any receptors. The lifespan for the gustatory cells is about 10 days. So every 10 days, we lose these receptors and replace it, just like the olfactory. We lose it and replace it. This is one of the fast turnover. Primary taste sensation. What are the primary taste sensation? And this is important. Sweet, salty, sour, and bitter. These are the primary. Okay? So remember those primary taste sensation. If I ask you, it's sweet, salty, sour, and bitter. These are the 4 primary taste sensations. There is another one, additional one that's not from the primary. There is just an additional one. It's just a special type that's called umami and water. These two are not the primary sensations. These are additional ones. So the primary are 4. Okay? And you need to remember this. Now we have also umami and the water. What's the umami? This is the chicken and beef broth and Parmesan cheese. Just this specific taste. If you are not able to taste the chicken broth or the beef broth or the Parmesan, if you cannot, it means you're lacking these gustatory or taste receptors. You don't have those, right? Water receptors are available in the pharynx. So we have 4 primaries and 2 additionals. 4 primaries that we need to know, and the 2 additionals. What are the 2 additional? Umami, and this is the special sense, okay, special taste for the chicken or beef broth and the Parmesan cheese. Just those 3. You don't have it, you're not going to be able to taste those 3. If you don't have it, this is just additional thing. It's not salty, it's not sweet, it's none of the primaries. It's additional one. The water is not even picked up in the tongue. It's picked up back in the pharynx. Okay? Same thing, they work by dissolved chemicals, just like the olfaction. So how the olfaction and the gustatory sensation or the taste sensation work? Dissolved chemicals. Dissolved chemicals in something that we're smelling, or dissolved chemicals in the food that we're eating. They work through chemically gated ion channels. If you still remember the ion channels, they work by physical distortion, or they work by chemical or electrical. This is the chemical one. The gustatory is one of the G proteins that respond to sweet, bitter, and umami stimuli. So it is a specialized G protein that responds in the receptor that respond to sweet, bitter, and umami sensation. At the end, there will be a neurotransmitter that's released, and it will follow the gustatory pathway. There is something called PTC, and PTC is something that if we put it like a drop or something on your tongue, you might be able to taste it or not. 70% of the Caucasian can taste it and 30% cannot. Okay, so this is something that can be lacking in some people. It's something inherited. And obviously, same thing, just like the olfactory receptors, in elderly, specifically starting from 50, everything deteriorates. Now the eye. We are going to discuss the eye in details and the accessory structures of the eye as well. The eyelid is one thing. We have have upper eyelid, lower eyelid. We have a superficial epithelium. There is an epithelium that is covering the eye, except which part is not covered by epithelium in the eye? What do you think? The cornea. If the cornea is covered by epithelium, you will not be able to see. And this is an abnormality, some people have it. That the epithelium is, if you open your eyes like this and look inside, this layer is called the epithelium. If it invade the cornea, you will not be able to see. So this is the epithelium. The eyelids are called the palpebra. If you still remember the levator palpebrae superioris, remember those muscles? So the eyelid is called the palpebra. And the function of the eyelid is it's protecting the eye. You keep blinking, opening, and closing your eyes to keep the surface lubricated and to prevent the dust from entering to your eyes. The palpebral fissure is between the upper and lower eyelid. This is your upper eyelid, this is your lower eyelid. The palpebral fissure is this opening in between those two. So you open and close the palpebral fissure. There is medial and lateral canthus, which are the angles of the eye. Which one is this? The medial, and this is the lateral. So obviously, the medial is closer to the middle line, right? And the lateral is away from the middle line for everything. Eyelashes, this is the hair that prevent the foreign bodies from entering. There are some glands also are called the tarsal glands, and these tarsal glands secrete lipid-rich product that help to keep the eye sticking together, sticking together. I don't know if you ever see somebody doing like this. They open their eyes and they close their eyes and they have hard time opening their eyes. If you ever see something like that, they have a problem in the tarsal glands. The tarsal glands are the glands that secrete in this margin, like this, the margin, and it's lipid-rich secretion, so it will always lubricate and prevent the eyelids from sticking together. If somebody's having a problem with that, less secretion, or it's inflamed or something, they close their eyes and they are having some sort of hard time opening or detaching the two eyelids. So this will be a problem with the tarsal glands. The lacrimal caruncle, this is a mass of soft tissue, and it will produce thick secretions. And this is usually when you sleep, when you get up in the morning, we usually do like this, right? When you wash your face, you remove this. This is something natural that's secreted by the lacrimal caruncle. When you look in the mirror, in the medial canthus, you will see this fleshy red swelling. If you ever see that before, in the right side, take a look at it next time. And this is for secreting these secretions that clean the eye, and it usually will accumulate in the medial canthus, and this is where you remove it in the morning or something. So this is the function of the lacrimal caruncle. We will see all of that when we go to the lab, but just understand the function. Usually the lecture is all about the physiology, and the lab is usually more about anatomy, 90% anatomy, 90% physiology. So we will stop at the conjunctiva, and we will finish the rest next Tuesday.